High blood pressure or hypertension is commonly referred to as the silent killer. It contributes to more than 400,000 deaths in the United States each year, and it impacts no other group quite like African American men, in whom high blood pressure appears earlier in life and is often more severe. Despite having the highest hypertension-related death rate of any racial, ethnic, or sex group in the United States, many African American men do not seek care for this condition. We sought to address this healthcare disparity by developing an effective and convenient blood pressure program for Black men. The question became, what do you do when you can't get people to go to the doctor? And our answer was, you bring the treatment to them. Healthcare outreach to barbershops has a long history, but it was largely untested as to whether or not it could improve chronic disease management. In this study, our principal investigator, Dr. Ronald Victor, linked health promotion by barbers with drug treatment by clinical pharmacists and evaluated the resultant efficacy in a cluster-randomized trial. After 6 months, we found that blood pressure reduction was 22 points greater in the intervention group. If this hypertension model of care were widely adopted and scaled nationally, it has the potential to not only save lives, but millions of dollars in heart disease and stroke care.
.9% sodium chloride, or saline, sometimes called normal saline, it's simple. It's composed of water and table salt. And the amount of salt that's in there, sodium chloride, the amount of chloride in there in particular is about 50% more than is in human plasma. And in contrast, balanced fluids like lactated ringers or plasmolite A or Normasol all are designed to have an electrolyte composition, including a chloride level that's much closer to that of human plasma. Our study compared balanced fluids with 0.9% sodium chloride for critically ill adults and found that use of balanced fluids rather than saline resulted in a lower rate of death, new renal replacement therapy, or persistent renal dysfunction. The administration of intravenous fluid is the most common intervention received by hospitalized patients, and so the implications for this study are pretty broad, that these two fluids, balanced fluids and saline, have been used for decades, and it's not been known whether there was any difference between them for patient outcomes. The fluids are nearly equivalent in cost, and so we think these findings of better outcomes with balanced fluid as opposed to saline likely mean that transitioning toward balanced fluids will have a benefit for patients across a wide range of diseases and severity of illness and across a wide range of settings. There are over 5 million critically ill adults admitted to intensive care units across the United States each year, the vast majority, 80–90% of whom receive some volume of intravenous fluid. And so these findings, because our study was so broad, likely apply to almost all of those patients, or the vast majority of those patients. What we saw in our study was that for every 100 patients treated with balanced crystalloids or balanced fluids instead of saline, one patient avoided death, new renal replacement therapy, or persistent renal dysfunction. And so when those numbers would be applied to 5 million critically ill adults and probably also to patients in other parts of the hospital, we're talking about potentially tens of thousands of patients who are spared death, new renal replacement therapy, or renal dysfunction each year in the United States alone.
I'm a pediatric endocrinologist by training, and during that time developed an interest in metabolic bone diseases in childhood. We've had a major interest in inherited forms of rickets. More recently, the exciting development has been the identification of a novel hormone, FGF23, that is the agent that mediates this phosphate homeostasis. We were able to demonstrate that inhibiting this FGF23 activity with this agent for this disorder could raise the serum phosphate level by adjusting the level at which the kidney would retain phosphorus. And there are potential applications in other phosphate-related diseases. The satisfaction of seeing an entire pathophysiologic system emerge out of multiple people's contributions to understanding it, I think it's an honor to be part of this group and more than excited to receive the award on their behalf.
The American Heart Association announced brand new guidelines that changed the stroke window from 6 to 24 hours. we're going to double the number of patients that we could treat under a 6-hour treatment guideline.
So the way that we used to take care of patients with ischemic stroke is it was very, very time sensitive. We were on the clock. And what that resulted in is a lot of patients not being eligible for any treatment for their stroke whatsoever.
We would never be able to treat patients out at 10 or 12 or 16 hours. Patients who wake up with a stroke are not a candidate for treatment, and patients who have a stroke far away are not a candidate. One of the Stanford faculty members, Mike Mosley, discovered that you could see the stroke evolving in real time with diffusion imaging. So this was a new MRI technique. So what we learned from that is that it's not all about time, that everybody's stroke grows at a different rate. So as we noted this, we said, well, this could be a real breakthrough for treatment, because it means that some of these patients who are coming late, maybe we can treat them. So we knew that we needed to automate this, and we wanted to call it rapid because it had to be quick. I can be at home, out of the hospital, a patient comes into the emergency room, they get scanned, and immediately, within minutes, the image shows up on my smartphone. I know right away, is this a patient that needs immediate intervention?
Hemophilia B is an inherited bleeding disorder that affects predominantly men. And the problem with hemophilia B is that patients are missing coagulation factor IX, which results in recurrent spontaneous or trauma-induced bleeding. The bulk of this bleeding affects joints that can result long-term in arthropathy. It can also affect other organs, including the brain, that can have really devastating consequences and can be associated with fatalities. The current therapies for hemophilia B involve recurrent exogenous factor administration intravenously. So you can imagine this involves significant planning of daily activities and can result in difficulties in adherence. We administered an adeno-associated or AAV vector to adult men with hemophilia B to deliver a factor IX gene to their liver so that they can then make their own factor IX protein. What we found is that the administration of the vector resulted in sustained levels of factor IX activity that were on average approximately 35%, so just slightly below normal levels. But what's really unique is that for the first time we achieved factor IX levels that were adequate to essentially eliminate bleeding events and eliminate need for factor infusions. The men on this trial are no longer having to administer factor, they're no longer having bleeding events, and this has made a dramatic improvement in their quality of life. Patients are reporting simple daily activities that we take for granted, shoveling snow, coaching their son's baseball team, going to dinner and dancing with their wife. They've now been able to do unencumbered by having to administer factor or be worrying about bleeding events. The main ultimate goal of gene therapy is to have a one-time disease-altering or curative treatment, and so far that's what we've essentially seen in these patients. Ultimately, if that carries forward, the idea would be that patients wouldn't necessarily need care.
There are 1 million miscarriages in the United States every year. Miscarriage is the most common complication of pregnancy. For women who prefer to avoid a procedure to complete the miscarriage process, misoprostol has been used, but it fails commonly. As many as 40% of women who use misoprostol to complete their miscarriage will need multiple doses and need to come back for the procedure they wish to avoid. Our study's findings show that the combination of mifepristone before the use of misoprostol increases the likelihood that a woman will complete her miscarriage process by 25% over the use of the single drug misoprostol alone, which is the standard of care. This innovation is going to help women to return to their fertility more quickly after the miscarriage process is completed, to return to their work and family responsibilities more quickly. This improves the care for patients who have suffered a miscarriage. Mifepristone is a highly regulated medication. The FDA approved mifepristone in a way in which it is not accessible to providers or patients in retail pharmacies. Given the restrictions, future studies can look at ways of implementing the use of mifepristone and misoprostol for miscarriage patients in the current regulatory environment. There may also be opportunities to further streamline the regimen and improve patient-centered miscarriage care.
The issue that we're investigating is human cancer and harnessing the power of the immune system to fight cancer. And we do that through genetically reprogramming and redirecting T cells against cancer. The issue is that T cells typically don't recognize cancer as something that's foreign because cancer arises from within. But now, with great technology, such as gene engineering, we can introduce synthetic molecules such as chimeric antigen receptors into T cells. The findings of this study really center upon understanding the determinants of successful and failed CAR T cell therapy. We've identified the pathways that are important to potentiate optimal CAR T cell function, and also some of the biomarkers associated with response. Up front, from a simple blood sample, we can predict with some degree of certainty which patients would have complete, long-lasting responses to the therapy. This would allow physicians to prescribe CAR T cell therapy to selected patient populations, and that would lower manufacturing costs because we wouldn't be manufacturing for all patients, only the ones who would benefit, until we can improve the therapy for all individuals, and also lower the risk of side effects in individuals who are not likely to respond to the therapy. The holy grail is to modulate all individuals' T cells to make the therapy work for all future patients.
Breathing dirty air is bad for human health, negatively affecting the heart, lungs, and brain. Many developing countries face very high levels of air pollution from a combination of man-made and natural sources. And surprisingly, we still know very little about what these high exposure levels mean for health, particularly where other threats to health, such as from poor nutrition to infectious diseases, loom large. How important is the harm caused by air pollution relative to these other factors? Answering this question is hard. So how can we get a more accurate picture of the problem? In new research, we combine air quality measurements obtained from satellites with household survey information on nearly 1 million births across 30 sub-Saharan African countries. All of this data allows us to separate the role of air pollution from the many other factors that also affect infant health. So far, we've made sobering discoveries. We find that exposure to microscopic particulate matter is responsible for more than 20% of infant deaths in sub-Saharan Africa, and that this exposure led to about 400,000 excess infant deaths in our 30 study countries in 2015 alone. Our research shows that the health burden of poor air quality is perhaps twice as large as existing estimates. Compared to other popular health interventions like vaccine and nutritional supplementation, modest reductions in exposure to particulate matter, as achieved by wealthy countries, would have major beneficial effects.
For patients with rare conditions, it can be incredibly challenging to get a diagnosis. Many are passed from one institution to the next without finding answers. In order to address this problem, the Undiagnosed Diseases Network was started in 2014, backed by the National Institutes of Health. The goal of the project was to bring together clinicians and researchers from across the United States to develop a model for diagnosis and to stimulate research. In our recent publication, we present the results from the first 20 months of the study. Over this period of time, 1,519 patients applied and 601 were accepted for evaluation. Of the 382 patients evaluated over this period of time, 35% received a diagnosis. 21% of those diagnoses led to recommendations to change therapy, and 31 new syndromes were defined. It's our hope that these results will support the adoption of some of the UDN's diagnostic approaches more broadly. Our findings support the fact that this model can lead to diagnoses and hope for patients with undiagnosed conditions.
Transthyretin amyloid cardiomyopathy is an underdiagnosed, life-threatening disease that occurs when transthyretin protein accumulates and forms amyloid fibrils inside the heart. Without treatment, patients live only 3 years on average after diagnosis and typically die from progressive heart failure. Until now, there's been no medical treatments for transthyretin amyloid cardiomyopathy. In our study, a phase 3 clinical trial, we tested a new oral medicine called tafamidis. The drug binds to the transthyretin protein and prevents it from forming amyloid. We assessed the efficacy, safety, and tolerability of tafamidis in an international, placebo-controlled randomized study. We used a novel statistical approach to study rare diseases, the Finkelstein-Schoenfeld method, increasing the sensitivity and power of the analysis of a small cohort while prioritizing the importance of mortality and also addressing morbidity in the primary endpoint. In the trial, tafamidis lowered mortality by 33% and cardiovascular-related hospitalizations by 30% compared with placebo. Additionally, tafamidis significantly reduced the decline in functional capacity and quality of life and was very well tolerated. Transthyretin amyloid cardiomyopathy has been considered rare because the diagnosis used to require a heart biopsy. But now, a simpler nuclear imaging technique allows doctors more easily to diagnose the disease. And with the advent of an effective therapy, tafamidis, the potential to dramatically improve the outcomes for affected patients is enormous.